So finishing up here with the cell wall, it is external to the cell, plant cell, gives it protection, helps it from absorbing too much water, and it is not found in animal cells. So animal cells do not have a cell wall, but they do have a sticky coat on the outside that's referred to as the extracellular matrix. So this outer sticky coat, because it's made up of mostly carbohydrates or sugars, allows cells to stick to each other, but is also involved in communication between cells. Now, cells also are linked together via cell junctions. So if you consider like this, your skin, you can gently kind of tug on your skin and you're not going to easily pull the cells apart, right? You're not going to tear the skin easily, cause bleeding, because your cells are linked together by cell junctions. So they help bind the cells together. And also some of the cell junctions allow for cell-cell communication, because some of the cell junctions are called gap junctions, and they allow, or they have little channels between adjacent cells that are linked together, and cells can communicate with each other through chemicals or ions that would rapidly move between cells. Okay, so the nucleus, the organelle of the nucleus, is the control center of the cell, housing the genetic material, the DNA. And if you consider all of the DNA that's in the cell, it's often packaged into discrete units or discrete structures called chromosomes that are really only visible during what's called cell division. But all a chromosome is a tightly packaged up region of the DNA that's in the cell. And the DNA in your cells is broken up into distinct regions called genes. So a gene is the information for a particular gene product, which is a protein. An analogy I sometimes use is that all of the genetic material in your cells is like the master cookbook. Like the cookbook with all of the recipes to make everything. All, the cakes, desserts, entrees, all sorts of things. And a gene is a specific recipe. Okay? So the master cookbook is just information, right? The genetic material, your DNA, it's just information. The recipes, genes, are just information to make a particular product, in this case, a protein. So the analogy with the cookbook is a specific recipe allows you to make a specific product, like let's say blueberry pancakes, okay? A physical thing. The DNA, the genes are informational molecules to make specific gene products, which are usually always proteins. And then proteins do most of the work within the cells. So the nucleus has a double membrane. That's referred to as the nuclear envelope, and material gets into and out of the nucleus via these openings called nuclear pores. So the DNA is long molecules and combined with proteins, like that little video showed you, to form a structure that's referred to as chromatin. Okay? Each chromatin fiber is a chromosome, and humans in our nuclei, we have 46 chromosomes, and throughout most of the cell's activities, most of the cell life, the genetic material is in the form of chromatin. Okay? The genetic material, the DNA, doesn't form chromosomes until the cell's getting ready to divide. Okay, let's get back to chromosomes, all right? So the nucleolus is a region within the nuclear envelope, within the nucleus, and it has the genetic information to produce ribosomes. And then the ribosomes leave via the nuclear pores, and then the ribosomes are outside of the nucleus in the cytoplasm. And then the ribosomes, once they're out here, those are the sites for protein synthesis, where proteins are made on ribosomes. So the ribosomes, the sites of protein synthesis. So the ribosomes make the physical proteins that remain in the cell, or proteins that are going to be part of the plasma membranes, or proteins that are going to be secreted outside of the cell. Okay? So it just depends on what specific protein is going to be made. And the way the ribosome makes the protein is it gets attached to the copy of the gene. Remember, within the nucleus is the genetic material, the DNA, and specific regions of the DNA, genes, are the information for a particular protein. Well, that gene gets copied. The copy of the information, something called an mRNA molecule, leaves the nucleus out of the nuclear pore, and then a ribosome can latch onto the copy of the gene, the mRNA molecule, and it's the copy of the gene, so it's also an informational molecule. The information on the mRNA molecule is interpreted by the ribosome, and what the ribosome does is it links the different amino acids together to form a polypeptide chain that will become a protein. So the ribosome is the site, the physical place where proteins are made. And ribosomes are organelles as well as the other organelles in the cell. A ribosome is one particular type of organelle. So how does the DNA get converted into a protein? Well, the entire DNA molecule has specific regions, genes, and when the genes are expressed, the copy of the gene gets made into the mRNA molecule. So remember, if the entire DNA molecule is the master cookbook, a small section, a gene is a recipe. So the mRNA molecule is the copy of the gene, copy of the recipe. So then the mRNA molecule exits the nucleus, latches onto the ribosome, where the ribosome can interpret, read the information on the mRNA molecule, and the ribosome links the appropriate amino acids together to form initially a polypeptide chain that will become a protein. Okay? I hope so. Okay, so within the cell, there is organelles that are membrane bound, that are part of the endomembrane system. So membranous system within the cell. Endo is internal. So the nuclear envelope is part of the endomembrane system, along with the endoplasmic reticulum, and there's the rough endoplasmic reticulum because it has ribosomes on its surface, and there's also the smooth endoplasmic reticulum that does not have ribosomes, and then lysosomes that bud off of the Golgi apparatus, which is also part of the endomembrane system, yes. And lysosomes are involved in breaking down things like bacteria that might get into a cell, and then the vacuoles that are used to transport material around the cell, and also vesicles, which are also used to transport material within the cell. So these are all membranous structures, and the membrane is made up of the same material primarily as the plasma membrane. It's phospholipid. So here it's showing you, or showed you a little minute ago, the pathway for protein synthesis. The genetic material within the nucleus, the gene gets copied, right? The mRNA molecule exits, latches onto a ribosome. The polypeptide chain is formed. It migrates through the rough ER, rough endoplasmic reticulum. It is then transported to the Golgi apparatus through a vesicle that pinches off the rough ER, latches onto the Golgi, dumping off the proteins in there. The Golgi apparatus modifies the protein, and depending on where that protein is supposed to go, a transport vesicle pinches off and can transport the protein to the membrane for export out of the cell, or transport the protein to the membrane so it becomes part of the membrane proteins, or the proteins can become part of a lysosome, which works to break down and destroy things like bacteria. And then those proteins are a type of protein referred to as digestive enzymes. So a lot of elaborate stuff going on in our cells. So the ER, endoplasmic reticulum, it's the main manufacturing site in cells for a variety of molecules. The rough ER is called rough because it is studded with ribosomes on the surface. That means protein synthesis. So the rough ER is primarily involved in protein synthesis. The smooth ER does not have ribosomes on its surface, so it is not involved in protein synthesis. It is involved mostly in making lipids and breaking down detoxifying toxins or poisons that might enter into the cell. Okay? So rough ER, ribosomes on the surface, that means protein synthesis. And then here you go, the proteins can be destined for export out of the cell, or the proteins can be destined for the plasma membrane, or the proteins can become part of a lysosome. Just depends on what type of protein is being made. And the transport vesicles bud off of the rough ER, and they transport the proteins around. So the smooth ER, no ribosomes, so no protein synthesis, producing mostly lipids like steroids, and helps certain cells in your liver break down and detoxify drugs and or toxins that you may inadvertently ingest, eat, or drink. Okay. So then the Golgi apparatus, it's often considered like FedEx in the sense that the Golgi apparatus, once it receives proteins, is able to modify the proteins and identify where they're supposed to go next. So just like FedEx gets packages and is able to ship them places, the Golgi receives and refines, modifies, will store proteins, and then finally distributes them to where they're supposed to go. Okay? And the way it does that is by a transport vesicle budding off of the Golgi, and will transport those proteins to wherever they're supposed to go. Exit out of the cell, or proteins that become part of the plasma membrane, or proteins that might be digestive enzymes and become part of a vesicle, part of a lysosome that stays within the cell, and lysosome destroys things like bacteria. So the lysosomes, they are membranous sacs of digestive enzymes. Enzymes are proteins, and they do things like break down biological molecules to help provide nutrients within the cell, destroy things like bacteria, and can even identify old and worn out organelles and destroy them and recycle those building blocks for other things. So a lysosome in a way is part of the immune system within the cell, and kind of a cleanup crew within the cell as well. Okay, vacuoles. So these are membranous sacs, like the ones that bud off of the rough ER to transport things to the Golgi, in which case they're transport vesicles, or vesicles that bud off of the Golgi and go to the plasma membrane. So again, another type of transport vesicle. They can be food vacuoles from the plasma membrane, and food can be absorbed that way into a cell via a vacuole that serves as a food vacuole. Contractile vacuoles in protists can help pump out excess water. So there are a lot of uses for vacuoles. In plant cells, the central vacuole holds things like nutrients and water and pigments and even poisons. And that can be a large space or volume within a typical plant cell. Okay, chloroplasts and mitochondria, we'll continue those on the next part of this lecture.